good morning to our Polish colleagues again, and also good afternoon to everyone here. So thank you, Anna, for coming. Here's Nasta, here's Ferna, Shania, and we have online Zisian and Wenbo, and also I welcome Olga, Mugur, Witold, and Jakub, and Zelasko. Sorry, Zelasko, by the way, if you can switch on your the video, I think that would be really great. And Jakub, you can now share the screen. Is he sharing? Yeah. Okay. Sharing. Sure. Because I made it in a way so that I have only videos, so it's perfect. So we have like two different recordings. So welcome everyone. So without further delay, Jakub, the stage is yours.
Great. Thanks and welcome again. So I will try to answer your questions. I try to put it chronologically. I hope that Ilya will administrate whole event because it is a new experience for me. So okay, let's try. Let's try it. So first question was about transition from Western diet, hippocampal OTP, NOR impairment to cholesterol and pregnenolone axis in
Jakub, sorry, but we do not see this slide. Because probably you see it in the presentation mode, and we see still only the first slide, like this.
First slide now.
Yes.
Jakub, I think you have shared the wrong screen. Try to stop sharing and find the screen where you have not presentation mode, not this additional supplement mode, but the different one. Or if you cannot do that, you can change in the settings of the PowerPoint to
I have it. Yeah, I have it. Now is okay.
Yeah, it's okay.
It's perfect. Super. Yeah.
Right. So again, the first question was about transition from Western diet to hippocampal LTP, NOR impairment, and cholesterol pregnenolone axis in RQ8, and it was somewhat abrupt. So what drove the authors to this direction? I put some information from the introduction, and pregnenolone and neurosteroid production and involvement in a range of neuropsychiatric and neurological disorders were reported previously. That's why they found this bridge between cholesterol metabolism and cognitive disorders. That's why they designed their experiments in this way. And for me, it was pretty logically, and I hope that it was the cue of this question. So yeah, in my opinion, it was logically, but you needed to check the introduction where they manifested that, and maybe it wasn't so clear in my presentation that pregnenolone wasn't something new. It was known previously that it has some kind of involvement in cognitive disorders. I can see you. Is it okay? Is it satisfying for you?
Yeah, it's okay.
It was my question. So for me, it was satisfying. Thank you very much. now
okay, great. Okay, second one was where is this? Oh. Okay. Sorry, I got some problem with changing slides.
No worries.
Okay. No, okay. So second one was a little bit more complicated. It was also from you, Ilya, I suppose. And it was about mechanistic links between metabolic stress, pregnenolone reduction, POMC neuronal activity reduction, hippocampal LTP reduction, and authors demonstrated causation of these processes, but they didn't provide mechanism for those causative links. So I found information, but in papers which they cited in this article, that mechanism of changing in POMC neuronal activity in RQ8 in effect of Western diet may be caused because of long chain saturated fatty acids compounds which are metabolized from this kind of compounds, such as ceramides, which produced some kind of pro-inflammatory effects in hypothalamus. So they try to said that Western diet may be cause of some kind of inflammation in hypothalamus, which may result in impairment of functioning arcuate neurons. And what's interesting is that pregnenolone, I don't have this information here, but it will be later. It has somewhat anti-inflammatory effects. And as you can see, it may be modulator of CB1 receptor. So it is also probably some point of attach for next mechanistic studies. But authors didn't mention, direct mechanism of these causations. So yeah, it is, in my opinion, this kind of situation.
Thank you.
Is it okay?
Yeah.
Great. Right. So next question. Which sex-head animals used in the study and why? So all studies were performed in males, and authors didn't explain that. But I suppose it was because of the changes in neurosteroids levels during ovarian rhythm of animals. In females, analysis of the results would be much more tricky than in the males. So I suppose it was that was why they used only males, just for for easier analysis.
For standardization, so to say. Yeah. Okay.
Yeah.
Thank you.
Great, thanks. Do you know the normal amount of pregnenolone that is present in the brain? So, firstly, I've got information from Wikipedia where you can find that it is even detected in circulating blood in sera, and as you can see, it is at pretty high level, but the range is also quite wide. In adults, it is also observed in children in the adolescence. What's interesting, okay, and analysis of the level in the brain is pretty tricky because the units which are using for presentation of data is different in papers. Here you've got information about nanomoles per kilogram of tissue, and it was in this units in human brains of not demented or Alzheimer patients and it was post-mortem sections. So you can see here the normal, let's say, amount. And let me show one more thing on the next slide. Because in next question, there was effect about similar effect, which will be achieved by administrating pregnenolone through the bloodstream. And there's information from rats. And as you can see, normal levels in different, okay, maybe I will put pointer. In different brain structures, it is in this way, and this is in nanograms per gram. Gram of tissue. So as I mentioned during my presentation, authors put 10 micrograms of pregnenolone to the arcuate. So it was huge dosage. And according to this question, you can see that administration of pregnenolone to intravenous may be also effective because the level of pregnenolone is increasing after, and it is crossing blood-brain barrier. But there was also even some kind of preclinical tests on patients with schizophrenia, and there was no beneficial effects on cognitive functions in schizophrenia. So probably intravenous infusion in humans wouldn't be effective.
By the way, regarding the different concentrations, you mean different units of concentrations, which you mentioned. You saw that in that paper, they used some normalization is per milligram of tissue, and for the Acute, they say per punch. Did you see that? So
yeah, exactly. It was like whole tissue.
Yeah.
And I think that's why they named it enriched arcuate or something. Enriched. So they were not even sure whether they punched the proper tissue. This is what I think, because otherwise I do not know why they say enriched arcuate.
And here, like, just to be like even more, picky, I would even ask whether they actually check written in the methods how large was the punch, because there are different punches of different sizes. So, but okay, so I'm not actually I'm not really interested into that, but I would be interested whether the authors were rigorous enough to show all these details here.
Yeah,
not a question, just a comment.
Yeah, cool. Okay, so is it okay? I suppose it was questions from Olga.
Yes, thank you, Kuba. It's okay.
Great, thank you. So we can go to the next question, in which that in the introduction part of the article, they mentioned about study as a potential marker of early stages of Alzheimer's disease. Can you give a bit information on how dramatic the decline in START1 levels is in AD? All right, so there is only citation that it is a possible marker. I checked this paper, which was Arenas et al., and they found some changes here in the STAR1 immunolabeled level of STAR1, and it is not so big because there are two samples. On red is Alzheimer's disease patients, and on black Down syndrome. And as you can see, it is in the post-mortem slices from patients. So in my opinion, it is not the best biomarker, because you can do the same with the amyloid beta. Yeah, and in my opinion, it's not a good marker. And on the other hand, it is not so related with paper, because it was like, I suppose it was like in the discussion and showing functions of STAR-D1. Is it satisfying or?
Okay, thank you. It was about early stage, and then you are comparing now the amyloid beta accumulation, but amyloid beta accumulation is not detectable.
It is in
the
hippocampal samples. It would be also observed in early stages, but I think it is.
Okay. Okay, thank you.
Okay, thanks. Next question was about sex differences in terms of pregnenolone levels. In that research, it looks that they only used male mice, and for humans, they used samples from both sexes. As I said, they didn't speculate why they did it. It was only mentioned in the methods that only males were used in the animal studies. But I also found information about changing in pregnenolone during estrus cycle in mouse. And as you can see, the change of pregnenolone is observed even in the same day. It is in the triangle. So there was on the analysis of the brains at the noon and midnight. So the differences during cycle is huge of this compound, of this neurosteroid or precursor of neurosteroids. So its analysis and reliable and replicable data obtaining from females would be really tricky in this kind of research, in my opinion. So I think that using males is good idea.
Kuba, and in case of humans, also only males were used?
No, it was both, but it was analyzing not in the specific brain region, but in CSF. And if you check the limitation of studies, they showed also information that it may be, it is not very direct answer about and mechanistic answer on the question of the pregnenolone function, because it is more let's say overall state of the brain, because CSF is very unspecific tissue. Yeah. Good. Are we going next?
Yes.
Good. Okay. As a comment, it would be nice to show whether the short or long-term Western diet induced decrease in pregnenolone levels is temporary or permanent. So in males, I suppose that diet is not an acute experimental factor. So these changes pregnenolone should be permanent, but the authors even didn't show the time where animals were sacrificed for tissues, and they put only one time point. So I suppose it is permanent, but maybe it's changing in males also. I don't know, but I suppose that feeding of diets even 4 days, it is pretty permanent state. It is not something really acute.
Maybe I would argue with that.
Okay.
I would say we don't know, because simply if it is not tested, then it could be permanent, but I would be really surprised if something would be really permanent in the biological system, where you have mechanism of compensation, some aggravation, and whatever. So yeah, I would say we don't know, right? So because they have only one time point, which is 4 days after the start of the treatment, I assume that at that point they perform their experiments, all these behavior experiments, and they sacrifice the animals in order to get the slices for these recordings. And then probably for some other mice at that same point, probably they also checked these concentrations, but they didn't tell about any other more prolonged effects. And that was probably also a little bit beyond the scope, right? Because you cannot address everything in one paper. But it would be safe to say that we don't know, right?
Yeah, sure. It is. You are totally right. But there is also this decreasing in the level of pregnenolone after long-term feeding, and it was pretty the same as after short.
That is true, but it was measured also immediately after this experiment finished. So they just had this exposure to the high-fat diet. We didn't have this washout period where the mice were not exposed to high-fat diet, for example, for months. And we don't know whether this would be reversed to the initial levels, or maybe not, or maybe slightly attenuated. We simply don't know. So it would be safe to assume that we just don't know, right?
Yeah, sure. I agree with you. Okay. Right. And it is the last question, and it was about targeting hypothalamic pregnenolone as a viable therapeutic strategy for improving recognition memory deficit associated with metabolic disorders, and what are the potential challenges in translating this research into clinical applications. Okay, so I found information that this compound was also using as anti-inflammatory compound in rheumatoid rheumatoid arthritis treatment in 50s of 20th century. And after that, there was some clinical trials, as I mentioned you previously, in schizophrenia medication and changing and let's say improving cognitive functions of the patients with schizophrenia. And there wasn't observed any beneficial effects of this compound. So, in my opinion, it wouldn't be effective in this kind of disorders. And as I mentioned to you previously, it is working probably only because of high doses injecting directly to the arcuate. So yeah, in my opinion, in clinical approach, it wouldn't be effective. But who knows? Yeah, I don't think so.
I would also maybe add here that since they really see the effect, which is not maybe specific, but it tends to have like more specificity towards Acute, and actually they even identify the specific population, the POMC neuron population. So if we would kind of speculate, and if we can just think about how would it look ideally, yeah, if we had a tool that we can make sure that we have the local increase of the concentration in the POMC, like in the vicinity of the POMC neurons, maybe, or in the POMC neurons somehow. And if we would have a possibility to have this tailored way to increase this concentration there, that could have been a possibility to test, right? Because otherwise, if we are constrained to those intravenous injections or even other systemic ways of delivery, even into CNS, this would result in huge concentration, which would probably go to the limit of the toxicity, which would be toxic for many different tissues and cells. So this is probably just not feasible at all in this way. So in this regard, probably I would agree with Jakub, so that we just don't see that even in the horizon. But in the perspective, who knows? Maybe indeed. So this POMC specific local concentration is indeed so potent that it might have this potential to really modulate these phenotypes, but we just simply don't have these kind of tools, I think. So
yeah, I really like your comment. It's really good. Yeah, if we will have perfect delivery system for POMC neurons in arcuate, I think that the next question should be to be sure that it is the pregnenolone effect, not any neurosteroid which is produced from that. So in the perfect way with perfect tools, we would use, I suppose, very specific compound, and it wouldn't be necessary pregnenolone.
Jakub, here I cannot agree more, really, because you're absolutely right. Pregnenolone just is a precursor of many neurosteroids, and which of those derivatives, yes, of those steroids is actually having this beneficial effect, it still remains to be shown, right? And how does it actually signal through these POMC neurons to the hippocampus? So I absolutely agree with you. So if to target some of those neurosteroids, then it would be safer for sure to target something which is like one of the derivatives, not the precursor of multiple of those, to limit those kind of side effects. I absolutely agree with you.
Yeah, great. So it was the last question. So now I don't know what to do. So I'm waiting for Ilya.
Now, Jakub, you can maybe tell us about your attitude, your kind of what do you think about this whole study, about how adequate was the design, how adequate were the hypotheses, how adequate were the methods which they used to address this hypothesis, and overall, like, did you like the study, or like what the problems did you find, like anything, any kind of critics, like positive, negative, everything, what you want to say, and maybe some kind of conclusion.
Great. So in my opinion, this paper was really good. I suppose that the review path was really long for them, because there was really a lot of different paths where they go with that. They went with this paper. I think that it was very logical. This experiment design and methods were really impressive. They used, they checked the projections of the POMC neurons. They used DREAD system. They did a lot of behavioral tests. Maybe it would be good to show different neurosteroids and its role in POMCs and Acute, but they used human samples. I think, yeah, I would be really, really proud if I would prepare this kind of paper. It was really nice, and I had a lot of fun with preparing presentation about it, because it was really complete, in my opinion.
Well, here I would
I
also
have also one comment,
Yes,
if I may add. So in general, we say a lot about the influence of the diets that we are exposed to, obesity, metabolic syndrome that influences our cognitive function. And there are not too many examples that really try to target this question mechanistically. And this paper is one of such examples. There are not too many really papers, attempts to show the mechanistic background of this relation. That's one my comment. The second comment is that it's very difficult to somehow prove it, and as you could notice, this impairment was a bit mild, in a sense that they detected it in the novel object recognition and call it very easily like an impairment in a cognitive function. The basis for this impairment in the novel object recognition may be of a different origin, not only the hippocampal, of course. And so it's a bit, how to say, overclaiming in my opinion, but still it was consistent in different models. And it was also consistent with the MMSE that was done in humans. Of course, the MMSE test is a very general one, and you really have to have the impairment in order to show some deficits. Because I'm not sure whether you noticed during the Jakub presentation that what kind of questions you have in this test. So they are really general, like what year do we have, where are you, what is the floor in the hospital or something, this kind of thing. So the fact that they detected these changes in these patients, it's really saying something. Okay, so that was my comment. Thanks.
Thank you very much, Vitek. Thank you, Jakub. I absolutely agree, actually, both with the Jakub's assumption as well as with the comments of Vitek. So, and actually, I also wanted to point to those details about how actually they discovered that and how they came up to this whole conclusions. But first, maybe somebody else would like to share his or her opinion. Anna, Nastya, Verna, Shania, or Mugur, Olga, Wenbo, you're welcome to comment. Okay, so while you are maybe thinking about like what would be interesting for you, so let me share some of my insights. So I'm really with Vitek here in order to really praise how these guys did it. They started with this really tiny difference. So they checked these two different paradigms, and they found this difference which was very which was just on the border of significance. Yeah, so they see that this discrimination index, so they don't see any change. They only have a trend of the exploration time, the total distance, but this discrimination index is the only one which they show as a significant. We don't know how many they checked, but this is the only thing which they go for. So I personally, if I have the data point, which is just proven by just one evidence, by just one kind of site, but one experiment, I would be very, very cautious with that. And I would like to praise these authors because they went with that, and they really tried to look into the mechanisms, whether this is really a true is it not just a statistical variation, is it not just by chance, and they really go through all of this until they really just find the whole story and just dissect the whole mechanism. So I really like this because
if I may add something. Sorry, it is also very interesting that they used only 4 days feeding of these animals. It is, it is so short time, and they observed somehow, you're right, that it is a really delicate and really small effect, but it is amazing that after 4 days, they observed some changes, in behavior of the animals.
Absolutely, and I of course understand the editors of the cell metabolism, why they really found that this is something really novel, something really link those metabolic effects and this the hippocampal related effects. And I agree with Vitek. Yeah, so it's even not really clear whether what they see, this effect, is related to this LTP in the hippocampus, or this link of the POMC to the hippocampus. Or maybe it's something in addition, but truly there is something going on, and these guys managed to grasp this. So maybe they grasp just the edge of that, or maybe that is actually the core. Maybe this is indeed this causative, the main thing which is happening, or maybe it's just a contributing. We don't know. But overall, I totally agree with Jakub. The data are very solid, even though in some other cases, if I would see the difference like that and showed only in one experiment, I would really be questioning that. But the point is that they really show it in different settings. They try to use all these different models in order to consistently prove that indeed something is really going on. So even uncovering it just a little bit, maybe not fully understanding the whole mechanism, this is really deserves the praising, because I agree. So that there are not many studies like that. I absolutely agree with that. So let me scroll a little bit further. Maybe I would because I thought I also had some other interesting points here. So yeah, so it's really nice, and I agree, Jakub. So just 4 days. So it is so interesting to see how does it really change.
If I may add at this point, I even thought that maybe they were explicitly asked by reviewers to show that they have the same kind of effects before the metabolic changes starts, right? Because they, at the same time, after 4 days, they do not observe anything in the glucose levels, insulin resistance, and things like that. So who knows? Maybe it was in the beginning. It was the first experiment was done after a longer time, and afterwards they were asked explicitly to use these 4 days just to have a short time before they have any metabolic changes.
You're absolutely right. So it could be that they have actually started with this long-term exposure. They found some interesting differences, and then then in the end they found this extraordinary that actually it even works after 4 days, maybe just by chance, and that's how they started the story. So it's not necessarily they wrote the story in the way how they actually found that. So I totally agree with that. So it could well be, yeah, indeed. So anything else interesting here? Let me look. Because I thought I also found something, some other interesting points here. So it's really yeah. So the whole since it is so multifaceted, right? So it has like multiple different sides. So it was clear that's why it was really targeting the right journal, so that the reviewers and editors were really glad to have that. But otherwise, like for some of those experiments, you can see, like if you just isolate, if you look into just one of the experiments in the isolated way, just one figure, you would say, okay, so those kind of differences are kind of they cannot be that they published in Cell Metabolism using this having such tiny differences, right? And yet. I totally agree. So that it is really a really great study here. Okay, anything else?
I have a question.
Yeah.
So you mentioned that they work with male mice. So if someone wanted to try to replicate the experiment with female mice, what kind of adjustments do you think they would have to do to the experiment or the biomarkers or anything specific?
Jakub, would you like to?
I think it would be even tricky to get statistically significant results of pregnenolone concentration in the brain. I think it would be really tricky to get mouse in the same point of ovarian rhythm, and I suppose it would be tricky to say that it is this stuff, because as you can see, neurosteroids and steroid hormones, because okay, it is not only neurosteroids, but it is also estrogens and androgens, and it would be really difficult, I suppose, to show these changes, especially on this hormonal level, because maybe this behavior of females is the same, but it will be more tricky to explain why and connect it with pregnenolone, in my opinion.
And that's probably the reason why still Cell Metabolism accepted this paper done only on males, right? So ovarectomy would be one option.
I also thought about that. Yes, I would agree. It could have been very messy. So with the females. But who knows? Yeah, so it could be that we have seen many phenotypes and many different studies where they show really huge differences in males and in females. Who knows? Maybe this effect could be even more robust and even more pronounced in females. Could also be. Yeah, so we just don't know. But it's a great question. So it's worth studying for sure. Yeah, absolutely. Okay.
It is also interesting. Oh, sorry. It is also interesting how this knockout of the STARD1 works in females, yeah, because hormonal system is much more complex in females, and it is one of the most important, I suppose, transporters for production of the hormones. So it may be also changing phenotype of the females.
Yeah, great point. Great point. Okay, any other comments? Verna, Shaya, Nastya, Anna, Olga, Mugur, or Vitek? Okay, if not, yeah. So then we can conclude today's meeting. Really great, Jakub. I found your presentation really clear. So I really like that you started with the graphical abstract. This is the way how I would also would do that, and this very structured, as the paper is, right? So this paper is also really nicely structured, really logical, and then just decipher this mechanism from like one step to another, and so on and so on. So yeah, really great paper and really great presentation. and also really great input from all of you. Thank you very much. So we'll see each other in 2 weeks, right?
Great, thanks. And I want to say also that I'm really glad that we are using the Zoom system. It works perfectly. Thank you that you improved that. And yeah, and thank you very much. It's much better than the last one in my perception.
I totally agree. I think it works really smoothly. Yeah. Okay, thanks for the suggestion, actually. Yeah. Okay. Thank you very much, everyone, and have a nice day.
Yeah, you too. Thank you. Bye.
Bye.
Bye-bye.